Great. Thank you so much to the organizers for the invitation and to you all for your attendance. It's my pleasure to share with you all some of our recent work in our group on protein generation with evolutionary diffusion. And this has been really a huge team effort across many researchers and scientists. And today we're going to take a dive into the ever-growing intersection of generative AI and biology, specifically in the world of proteins. So I'd like to start with this visualization, which takes us down into that nanoscale world, where here what we're seeing is a visualization of a single membrane boundary in the nucleus of a single cell at 1 million times magnification. And these individual molecules that are illustrated and colored are, in fact, proteins. And as we know, proteins are biology's actuators. They underlie and execute all of biological functions. And because of how critical proteins are to every process in biology, from how we fight disease to how our cells get nutrients, there's this incredible opportunity motivated by the question of, can we design new proteins to unlock new biological and therapeutic functions? And this is exactly the grand research challenge that motivated our work, where our vision is to build generative AI models to achieve functional protein design. Imagine the ability to specify a language prompt, say, design a new protein that targets breast cancer cells to help deliver drugs, and having a generative AI model that can create specific designs for candidate proteins that would meet that function. This generative ability would unlock tremendous opportunities across applications, sectors, and industries relevant to the molecular world, from how we design new proteins as new molecular tools, like CRISPR-Cas systems for genetic engineering, to engineering new proteins and enzymes that can perform new chemistry that's not seen before in nature, to eventually designing new therapeutics for diseases like cancer, autoimmunity, and beyond. And to really tackle this question, we have to go back to thinking about how we can understand how to define and represent a protein such that we can build AI models to now understand and design proteins. And as we know, every protein is defined by a sequence of amino acid chemical building blocks, what we can think of as a biological language of sorts. And this sequence of amino acid building blocks can then form a three-dimensional structure that we traditionally then think of as performing or executing the function of that protein. And as we know, there's been tremendous research in this space that tackles parts of this process from a computational perspective, building AI models of representing sequences, or building models that can generate new structures, or the development of tools like AlphaFold that predict structure from sequence. Our goal in this work was really to turn this paradigm on its head, with the vision of going from a desired function to a new protein sequence that could then be synthesized, tested, and used. And we designed a generative model called EvoDiff that gives us a foundation for doing exactly this, by learning from large-scale protein sequence data to generate new proteins, and eventually moving towards the ability to be able to prompt the model with desired function to meet desired goals. And indeed, our model that we call EvoDiff is a diffusion-based generative model to achieve protein design. We've trained EvoDiff on large-scale protein sequence data that spans what we think of as evolutionary scale, meaning proteins and sequences that have been taken spanning across different biological functions from different organisms from across the tree of life. And after training, we can then use our trained EvoDiff model to generate new protein sequences that hopefully will meet desired function. And this was work that was a huge team effort from across researchers and scientists and interns from MSR, and really spearheaded by an applied scientist in our group, Sarah Alamdari. And I'll focus the talk today on sharing the backbone of EvoDiff, our model, and some of our recent empirical results in terms of quality of protein sequence generation that we're able to achieve with EvoDiff. To get into the modeling framework and the background for our model, the backbone to EvoDiff is a diffusion model, which, as you probably know, is a very, very powerful class of generative AI model that basically is able to learn to generate new data from noise. And indeed, in case you're not familiar, the most powerful models for image or video generation, like DALL-E, use diffusion models as the underlying backbone as a mechanism to generate new images. And the intuition and principle behind diffusion models is really beautiful and really simple, in my opinion. And what these models do is, in training, they look at consecutive instances of data, let's say images in this case, that just differ by the addition of incremental amounts of noise. And the objective, the task that these diffusion models are trained on, is to learn how to remove this noise step by step to get back to data, such that at inference time, at test time, brand new images can be started, brand new data instances can be generated, starting from an all-noise state. With the EvoDiff, we take this same principle and now apply it to the space of protein sequences. EvoDiff learns to generate new protein sequences in a similar way. But importantly, unlike images or other forms of continuous data, protein sequences are inherently discrete. And so to train EvoDiff, we have to come up with a way to noise a protein sequence. We do this by taking that amino acid sequence of representing individual chemical building blocks and corrupting the individual tokens, the individual residues in those sequences with a special corruption token. And importantly, unlike a left to right traditional autoregressive model or a masked language model, with which diffusion models are closely related, this corruption that we're performing in EvoDiff happens in any and all possible orders, across all possible orders within a sequence. We do this over a fixed number of time steps to be able to reach the point of complete corruption and take these incremental examples as now training data with which we can train EvoDiff. EvoDiff learns how to undo this corruption step by step to be able to now generate new proteins starting from an all masked, all corrupted state. In our work, we trained our EvoDiff sequence models on a data set of approximately 50 million unique protein sequences using a 640 million parameter model as the backbone architecture to learn how to learn this neural network denoiser that can remove this corruption step by step. What's beautiful then is that after having trained EvoDiff at inference time, we can then start from a all masked, all corrupted sequence of a specified length, and the model then at inference undoes this corruption step by step. And so what you're seeing on the left is a visualization of how this generation can occur in sequence space, and on the right is a pure visualization of the likely predicted structure that could result from the generated sequence generated by EvoDiff that I'm showing you here on the left. And so really the core to EvoDiff and to our framework is these two components. The first being this discrete diffusion modeling framework for learning and generating new protein sequences. And the second being the fact that we train on an evolutionary scale data set of sequences that span organisms and functions of proteins from across the tree of life. Now, transitioning to how we think about putting these two together to generate new protein sequences and actually evaluate the quality of these generations. In our first round of evaluations, we focused on 3 axes of metrics and 3 axes of criteria that we wanted to evaluate EvoDiff by. The first being the plausibility, the quality of individual generated sequences in terms of their stability, likely structural stability, and likely biological plausibility. The second being diversity, how different are the generations that we're producing, how well do they cover the distribution of natural protein functions and structures that we know to exist in nature. And finally, we also wanted to test the ability of our framework, at least in a preliminary sense, to enable that vision of controllable or conditional design. And so I'll touch on each of these three fronts in sharing some of our empirical results with EvoDiff. So first, considering the plausibility, the biological plausibility of these generations. We began by taking our trained EvoDiff model and using it to generate new protein sequences. And what we did here in this workflow is we generated 1,000 distinct protein sequences. And for each of those 1,000 generations, we wanted to evaluate the quality of those individual proteins in terms of their structural soundness and plausibility. So we comprised this workflow in which we took those generations. Again, remember that this is amino acid sequences. We predicted the likely structure corresponding to that sequence using a structure prediction tool like AlphaFold, evaluated the confidence of that structural prediction, and then also performed a step of, if we now inferred an amino acid sequence likely to fold into that structure, how closely does that prediction match to our original generation? And so here, what we're visualizing is that relationship between that structure prediction confidence and that consistency of self-folding for first natural proteins from the test set. And when we visualize the generations from our model EvoDiff, we see that we can span the distribution of structural confidence and self-consistency relatively well. And importantly, that we result in generations that are highly structurally confident and self-consistent. And we can also visualize the individual generations, select examples that were handpicked across different sequence lengths to verify that EvoDiff is in fact generating plausible and structurally sound proteins from an unconditional perspective. And indeed, currently we're in the midst of taking some of these generations and also validating them structurally, experimentally in the wet lab. We also wanted to consider how well, from a diversity perspective, these generations were able to capture the natural distribution of functions and structures that we see in nature. And so here we performed a distributional analysis where we took 1,000 sequences from the test distribution, used an independent language model, ProT5, to generate functional embeddings for those sequences and visualize them here in 2D space using UMath. We performed a similar type of analysis now for generations, 1,000 generations sampled from EvoDiff, taking the resulting embeddings and overlaying them in our visualization with those from natural test sequences. And we see we get decent coverage of this space for samples from our EvoDiff model relative to the span of the functional distribution of natural proteins. This is all well and good, but I think it's best contextualized first quantitatively by measuring the closeness of these distributions, where a lower score, a version of a Fauché score, indicates a closer match, but ultimately by comparing the resulting coverage of generations from EvoDiff with other approaches for protein design. And namely, when we look at a autoregressive left-to-right language model, we see we enable comparable performance in terms of coverage of this functional space, But as we'll see in a little bit, EvoDiff has this ability to enable arbitrary conditional generation, while the left-to-right autoregressive model is less flexible in this sense. Similarly, if we now compare to samples from a masked language model, like the protein model ESM, we see that the generations are biased towards a specific subset of this functional space, which we can attribute to the fact that EvoDiff achieves higher quality generations due to explicitly being trained for a generative task. And finally, we can also compare to inferred sequences from generated structures from a structure-based generative model like RF diffusion. And we see again that there is a biased coverage towards a specific subset of this functional space, which we can hypothesize can be due to the fact that EvoDiff is looking at a much larger training set that spans evolutionary scale, whereas structural data only exists for a subset of these proteins in this space. We can perform a similar analysis more explicitly from a structural point of view, again, thinking about how our generations cover the space of natural secondary structures seen in natural proteins, where again, we're performing a similar analysis across structural classifications on a residue-wise basis from the model ProTrans. And we can see that comparison of the distribution of inferred secondary helical structures versus strands for sequences from the validation set versus generations from EvoDiff are closely matching both qualitatively and quantitatively. And when we compare to autoregressive generations from a masked language model or purely structural model, we see that EvoDiff is able to better recapitulate the distribution of secondary structure elements that mirror those of natural proteins. So from these two perspectives, we have a sense now that generations from EvoDiff are both plausible and structurally sound from an individual level, as well as diverse and span the distribution of functional and secondary structure elements from a distributional and larger scale level. Our last thing that we wanted to highlight and showcase with EvoDiff was its flexible capabilities to enable conditional protein generation in a very flexible manner. The beauty of this is that most naturally, with this EvoDiff diffusion framework, we have a very natural way to automatically perform conditional generation. Because we perform this discrete diffusion modeling where we mask and corrupt sequences in an arbitrary and order agnostic fashion, we have the ability to do conditional generation by simply taking an arbitrary subsequence within the overall amino acid sequence, fixing it, and in-painting the remainder of the sequence by simply masking the desired portions and decoding those desired portions in any arbitrary order. And so you can see this come to life across different use cases for conditional protein generation using this paradigm, where we can think about looking at alignments of sequences and effectively masking just a single sequence in that alignment, or even looking at masking arbitrary portions which may be discontinuous within a sequence or within an alignment, and performing in-painting or scaffolding type generations to now condition a new generation on that fixed information. And so to give you a taste of how these conditional generation abilities work, one thing that we did in our paper and in our EvoDiff modeling efforts was to more explicitly leverage evolutionary information using this conditioning scheme. And to do that, we constructed a model that we call EvoDiff MSA, where we can take a single protein sequence that we call a query sequence, and perform a a homology search using a tool like BLAST or using other tools and data sets out there to now construct an alignment, an MSA of multiple evolutionarily related sequences related to that reference query sequence. We can then take that as starting data and using the same principle of this corruption and order agnostic masking, train a resulting diffusion model that we call EvoDiff MSA, where now we can, again, mask and corrupt arbitrary portions in now this multiple sequence alignment of several related proteins and train a model that can now generate entire MSAs at inference time, following training on these large data sets of multiple sequence alignments. And what's very beautiful about this framework is that effectively by performing this training over multiple sequence alignments, we're amortizing the cost of building a local generative model into that conditional information that's specified by this evolutionarily constructed MSA. And so a local generative model would work by taking sequences from a subset of a related protein family and training a bespoke generative model on that family. EvoDiff MSA gives us the ability to use the conditioning afforded by this diffusion framework to now perform evolution-guided generation in an amortized fashion. And this works by supplying that multiple sequence alignment as a form of conditioning information to EvoDiff, and then simply masking that first query sequence and asking the model to now, given the context of the rest of the MSA, to now generate a reference query as at inference time, amortizing the cost of that local generative model. And so more details on this and some of our empirical results are in the paper. But I think what was most powerful about this, the work and the results that we showcased, was our ability to take the EvoDiff sequence model and the MSA model and deploy it for functional tasks. And so one example of a task that we showcase is this notion of motif scaffolding. And this is motivated by the fact that in biology, many protein functions are mediated by a functional motif within that protein that's stabilized by a larger structural scaffold. And traditionally, this type of design task has historically been pursued purely through structural approaches. And so we asked, can we perform this same type of functional motif scaffolding with EvoDiff using only sequence information, no notion of explicit structural information? And the answer to that question is yes. And I think this is what is most powerful about EvoDiff in its ability to naturally enable controllable design through this conditioning via kind of inpainting, if you will. And so in this visualization, we can see that come to life in this example, in this context of functional motif scaffolding. And so here we've taken a protein, which is calmodulin, a calcium binding protein. And in green, we've annotated the motif, the part that's responsible for that calcium binding function. Using this conditioning capability, EvoDiff can take that functional part as a sequence context or a sort of biological prompt, and now design at inference a brand new protein sequence around that part, step by step, using the decoding framework from the bottom up. And what's amazing is now that this new generation, where you see that generated sequence at the bottom and the predicted structure at the top, shows a structure that we are now experimentally linking to function in experimental tests and validations. And so we can think about now applying this and extending this through a range of tasks, benchmarking the success of our method across a range of these types of scaffolding problems. And we see that we can perform quite well with both our multiple sequence alignment method and our single sequence alignment method, getting close to the performance and matching the performance of, in terms of number of these scaffolding problems solved relative to a pure structural based method. And again, the strength and the beauty of this approach is that there is explicitly no structural information needed to perform this kind of task. It's explicitly done purely through sequence modeling and purely through the power of this evolutionary scale training. And so with that, I'll conclude, and briefly to summarize, what we think of as EvoDiff gives us what we think of as a foundation, a first step, and as a platform to work towards this broader vision of controllable protein design and protein sequence generation, not only enabling capabilities for unconditional generation, but allowing for functional design and scaffolding through this ability to fix and inpaint arbitrary subsequences within a sequence, and now working towards more general capabilities in being able to specify through text or other modalities greater controllability and conditioning properties. And thank you so much for your attention, and happy to use the remaining few minutes, if possible, for any questions. Thank you.